Hi, in this session I am going to talk about pattern recognition. The pattern recognition is very much related to the predictions because once we able to find the pattern which in a sequence or in a structure, we can actually make prediction about the structure or the function of the macromolecule. So there are different methods for the pattern recognition, and here are the three major methods used in statistical pattern recognition. First one is classification. Classification is a supervised machine learning in which we are clustering or we are actually classifying the data based on some predefined features. Secondly, the dimensionality reduction, which can be a supervised or unsupervised machine learning, in which the features which is not essential, which is like actually associated with the data is omitted, so that the data will become more sharper and it will become the process of comparing or recognizing the pattern will become more easier. And finally, we have the clustering. Clustering is a unsupervised machine learning in which we do not have any features for classifying or clustering the data, and all this clustering is based on the properties of the data. So we are going to talk in detail about each of these methods for pattern recognition. First one is classification. The classification assigns features to predefined classes based on its features. So it analyzes the association between the features from the training data and to classify the unknown variable. So there are different techniques which can be used for classification, which include decision tree and linear regression. In all of these classification techniques, we have the training data and test data. The training data is that which comprises of representative data of a known category. The test data is an unknown category. So we use the feature extractor to extract the features from the input data, and this based on these features we are actually classifying the data into different groups, and the prediction may leads to the identification of the normal or disease condition. So once we made the classification based on the predefined features, if our test data is belong to one of this group, and if that group is associated with a disease condition or a special condition, there are probability that our protein or our DNA sequence is associated with that gene. So like that we can make predictions. So it's all about we are classifying the data based on the features. Okay, if our test data is belongs to one among this group, features of that particular group is also applicable to their test data. Okay, so suppose if we have five different variables of a gene, and each of these variables is associated with one or other condition. Some of them will be normal, some of them will be affected, and if we get a novel DNA sequence from a patient or a suspect, our DNA sequence is belong to one of this group. If that group is associated with a disease condition, it is possible that our sample data or test data, or the person from where we got the samples is also affected, or there is a chance of getting affected by that disease condition. So that's how the classification works. So first we classify all sequences, and then we looking for the test sequence and whether it is belong to the which category. Secondly, the dimensionality reduction, which is actually a method to store the data in a compressed form. So what we do, we actually minimize the number of features associated with the data. Okay, there will be different features. If you take the microarray data, there will be different features and different functional annotations which is associated with the data. What we really do is that we will omit or we will avoid the features which is not essential for the this comparison or the recognition, and we will only take the important features along with the data. So one major method used for dimensionality reduction is PCA, principal component analysis. In this, the data set with the p variables are reduced to a smaller set with a k variable. So p will be a huge number. Let's say the p variable one data or microarray data is associated with 1,000 different features. And in this PCA, what we do, we will avoid the non-important features, or the features which is not relevant to our study. So we'll be reduce the 1000 into 100. So at the end, our data, or our particular microarray data, is associated with only 100 features. So 100 important features. So it will become more easy to compare with the other, or do the analysis. Uh, it is especially useful for determining features, spare features representation. It can be done either by discarding irrelevant data, or combining the relevant measurements. Okay. So the feature selection, or the dimensionality reduction, will increase the accuracy of the prediction, because we are avoiding all non-important features, and it will reduce the complexity of the process. It will make interpretation more easier, and we can use different methods for selection of these features, which include filter, wrapper, and embedded method. In filter method, what we do, we are giving score for the each features, how much relevance this feature is having, or this particular sequence is having. Based on the relevance, we are giving scores. So that's how the filter method work. In wrapper, we are combining it into subset, and we are the subset are combined, like actually compared and evaluated with the other combinations. So we'll get the best combination out of this different subset. In embedded method, we are using the modeling of this computer aided modeling for the selection of the features. So in dimensionality reduction, what we really do, we are reducing the number of features associated with the data. Okay. When the features are, number is high, it is very difficult to compare. If you are avoiding the non-important, or not related features, the number of features associated with the data will be less, so we can easily compare with the other data set. And finally, we have the clustering method. It's actually an exploratory data analysis technique, in which we are grouping similar features group in particular group. But when comparing with the classification, in the classification we know we already have the features groups, like we are making groups based on the features, and we already know which features belongs to what group. But in clustering, there is no a priori knowledge is available. Based on the characters within a set, we are actually classifying the data, or clustering the data. The clustering can be done by using two different algorithms: hierarchical clustering and partitional clustering. So hierarchical clustering build clusters by merging together or dividing data in different steps. Either we merge together similar data together, or we can divide in multiple steps in a hierarchical order. That's why it's called a hierarchical clustering. In the partitional clustering is done iteratively, but the number of clusters needed can be determined. So these are the methods used for pattern recognition. Thank you so much.